Hello, greetings, and good day to everybody. I am Professor Jun Cajigal and I hope all of you are doing fine. Now, this is the second installment of our online lecture about photosynthesis. Now, if you will recall, in our first video, we focus on the overview of photosynthesis, plus we have discussed the biochemical pathways involved in the light reaction of photosynthesis. Now, if we have any questions or even comments or reactions about the first video, please feel free to type in your questions, your comments, or your reactions in our comment section below. So before we go into the details of our main agenda today, which is the Calvin cycle or the dark reaction, let us have a quick review of the light reaction of photosynthesis. Now, in order to do that, we will make use of this table showing the comparison between the two reactions involved in the light reaction. We have the non-cyclic photophosphorylation and we have the cyclic photophosphorylation of the light reaction of photosynthesis. Now, everybody, let's take a look at this table. So for non-cyclic photophosphorylation, the electrons do not come back to the same molecule, and we started with P680 if you will recall in non-cyclic photophosphorylation. For cyclic photophosphorylation, electrons come back to the same molecule. In this case, the starting molecule is the reaction center P700 in photosystem 1. Now, for non-cyclic, the first electron donor is water, while for cyclic photophosphorylation, the first electron donor is the reaction center of photosystem 1, which is P700. For non-cyclic, both photosystem 1 and photosystem 2 are involved, whereas in cyclic photophosphorylation, it is only photosystem 1 that is involved. In non-cyclic, the last electron acceptor molecule is NADP, while in cyclic photophosphorylation, the last electron acceptor is P700 in photosystem 1. And lastly, for non-cyclic photophosphorylation, the net products are ATP and NADPH. Both ATP and NADPH would be used to power the Calvin cycle or the dark reaction. And simultaneous to that, oxygen is released as a waste gas. While for cyclic photophosphorylation, the product is ATP only. Again, if you will recall, some of the electrons from ferredoxin goes back to the electron transport chain so that more ATP can be produced by cyclic photophosphorylation, because the ATP for non-cyclic photophosphorylation is not enough to power the Calvin cycle or the dark reaction. So that's how we compare non-cyclic photophosphorylation and cyclic photophosphorylation, which are the two biochemical pathways of the light reaction. Alright, so with this, let us now go into the details of the Calvin cycle or the dark reaction or the light independent reaction. Light independent because this reaction can still proceed even without the energy from the sun. Now, here are some key points that you need to understand so that you could more appreciate the Calvin cycle or the dark reaction. Alright, so number one, as we mentioned in the first video, that ATP and NADPH produced by the light reactions of photosynthesis would be used to power the Calvin cycle in order to reduce carbon dioxide to sugar. So here, carbon dioxide will enter the reactions we will discuss later. And second point is that the Calvin cycle is similar to the Krebs cycle in that the starting material is regenerated by the end of the cycle, as we will show later. The third point is that carbon enters the Calvin cycle and leaves as sugar. Then point number 4 is that ATP is the energy source, while NADPH is the reducing agent that adds high energy electrons to form sugar. And the last point that you need to understand about the Calvin cycle is that the Calvin cycle actually produces a 3-carbon sugar glyceraldehyde 3-phosphate or PGAL. So those are the key points that you need to know about the dark reaction or the Calvin cycle. Now, the Calvin cycle is divided into 3 steps. So let us now focus first on step number 1. Now, in step number 1, carbon dioxide enters the reaction. Recall, carbon dioxide comes from the atmosphere, which enters the leaves of the plant through tiny openings called stomata. Now, the carbon dioxide reacts with a substance called RUBP. RUBP is the acronym for ribulose bisphosphate or ribulose diphosphate. The reaction between carbon dioxide and RUBP is catalyzed by an enzyme called Rubisco or RUBP carboxylase. Now, the reaction will cause the addition of a carbon to RUBP. Now, RUBP is a 5-carbon compound, as you could see in this diagram. Now, RUBP is a 5-carbon compound, plus you have here carbon dioxide. So what happens here is that RUBP carboxylase or Rubisco will fix the carbon from carbon dioxide to RUBP, forming a 6-carbon compound which is unstable. This unstable compound will easily split into 2 molecules of PGA. Alright, so that is step number 1. Alright, so again, carbon dioxide enters the leaves of the plant and reacts with RUBP or ribulose bisphosphate or ribulose diphosphate. The reaction is catalyzed by an enzyme called Rubisco or RUBP carboxylase. So what happens here is that the carbon from carbon dioxide is fixed into RUBP. So RUBP is a 5-carbon compound plus carbon from carbon dioxide. You form a 6-carbon molecule that is unstable and will split into 2 molecules of PGAL. So that is step number 1, which is also known as carbon fixation. Alright, so that's step number 1. Step number 1 is carbon fixation. So again, let's take a look at this diagram showing step 1 or carbon fixation. So here, carbon dioxide enters the plant through tiny openings in the leaves called stomata. This carbon dioxide reacts with RUBP or ribulose bisphosphate. Now, the reaction is catalyzed by an enzyme called Rubisco or RUBP carboxylase. So what happens here is that the carbon from carbon dioxide is fixed into RUBP. RUBP has 5 carbons in its molecular structure, plus you have the carbon from carbon dioxide. You form a 6-carbon compound which is unstable. This unstable compound will easily split into 2 molecules of PGA. or phosphoglyceric acid. Alright, so at this point, let us now summarize step 1 or carbon fixation. So what happens in carbon fixation is that this phase begins when a carbon dioxide molecule is attached to a 5-carbon sugar, which is ribulose bisphosphate or ribulose diphosphate. Then this reaction is catalyzed by an enzyme called RUBP carboxylase or Rubisco. And Rubisco is one of the most abundant proteins on the planet. And the products of this reaction is an unstable 6-carbon compound that immediately splits into 2 molecules of PGA or phosphoglyceric acid. Well, I hope everything is clear about step 1 or carbon fixation. So let us now proceed with step number 2. So let's take a look at this diagram. So we start with PGA or phosphoglyceric acid. Now PGA reacts with NADPH from the light reaction, and also PGA reacts with ATP also from the light reaction of photosynthesis. Now since NADPH is involved, we expect that there is redox or reduction oxidation. So we can call step number 2 reduction. Alright, so exactly what happens in step number 2. Now in step number 2, there is an enzyme that will phosphorylate or will add a phosphate group to PGA by transferring a phosphate from the ATP molecule. The product is bi-PGAL. Then the hydrogen from NADPH reduce bi-PGAL to form PGA. So what happens here is that NADPH is oxidized while bi-PGAL is reduced to form PGAL. So that's what happens in step 2 or reduction. So I hope everything is clear about reduction, which is step number 2. So again, PGA reacts with NADPH and ATP from the light reaction. Now let's take a look at this diagram again. Now what happens to PGAL or phosphoglyceraldehyde? Now actually PGAL is the final product of the Calvin cycle. But again, PGAL is really useless for the plant. So what happens here is that PGAL is further converted into sugar, and with the reaction of ATP from the cyclic photophosphorylation process, PGA is converted to RUBP. And then RUBP reacts again with another molecule of carbon dioxide, and the cycle continues. So this is step number 3, or we will call this regeneration of RUBP. Alright, so what happens now in step number 3 or regeneration of RUBP? So there is a complex series of reactions that rearranges the carbon skeletons of 5-glyceraldehyde 3-phosphate or PGAL into 3 RUBP molecules. Then the reaction requires ATP molecules from cyclic photophosphorylation, and RUBP is thus regenerated to begin the cycle again. Alright, so basically that's the Calvin cycle or the dark reaction. Alright, so again, here is a diagram that will compare both the light-dependent reaction and the light-independent reaction. So in the light-dependent reaction or the light reaction, light is needed to power all the biochemical pathways involved, plus water enters the reaction producing ATP and NADPH. Now, the ATP and the NADPH would be used to power the Calvin cycle, and at the same time, oxygen is released as a waste gas. Then eventually carbon dioxide enters the Calvin cycle and it will undergo a 3-step biochemical pathway. Like in step 1 we have carbon fixation, step 2 is reduction, and step 3 is regeneration of RUBP forming sugar. Alright, so that's the photosynthetic process undergone by plants or autotrophs. Alright, so at this point, let's watch this short video about the Calvin cycle. Alright, let's watch this.
That carry out photosynthesis. If we zoom in on this photosynthetic plant cell, we can see the chloroplast, where the reactions of photosynthesis occur. Photosynthesis consists of 2 primary steps, the light reactions and the Calvin cycle reactions. In this tutorial, we'll focus exclusively on the Calvin cycle reactions. The overall purpose of the Calvin cycle is to convert carbon dioxide from the atmosphere into carbohydrates, or sugars, which the plant needs to power its cellular activities and build new plant structures. The Calvin cycle reactions occur within the stroma of the chloroplast. The reactions use the ATP and NADPH produced by the light reactions to convert the CO2 to carbohydrate. Let's zoom into the Calvin cycle to take a closer look. The Calvin cycle could be divided into 3 phases: carbon dioxide fixation, carbon dioxide reduction, and regeneration of RUBP. The first phase, carbon dioxide fixation, basically captures the CO2 from the atmosphere so it can be used in the reactions. To do this, CO2 is attached to RUBP, a 5-carbon molecule. The enzyme used in this reaction is called ribisco, and the result of the reaction is an unstable 6-carbon molecule that quickly splits into 2 3-carbon molecules called 3-phosphoglycerate, or 3PG. Next, the process of converting the CO2 to carbohydrate begins. This phase is called the carbon dioxide reduction phase because we're adding electrons and energy to the CO2 molecule. During this phase, a sequence of reactions uses NADPH and some of the ATP from the light reactions. These molecules supply the needed electrons and energy for CO2 reduction. Electrons are added from NADPH, and through a series of reactions, 3PG is reduced to form G3P, a carbohydrate. ADP and NADP+ return to the thylakoids to be converted back to ATP and NADPH by the light reactions. One of the G3P molecules is set aside to be used as a building block for glucose, but the majority of the G3P molecules move forward into the 3rd phase of the Calvin cycle. In this phase, ATP is used to combine the rest of the G3P molecules to form RUBP molecules. This RUBP can then combine with additional carbon dioxide molecules, continuing the carbon reactions. To form a glucose molecule, the cycle actually has to turn 6 times, because each turn of the cycle adds only 1 carbon atom from the incoming carbon dioxide. It's important to remember that the Calvin cycle is dependent on the light reactions to provide NADPH and ATP, which in turn provide the electrons and energy needed to form carbohydrates.
Alright, so I guess that's about it for the Calvin cycle. I hope you have learned a lot about that video about the Calvin cycle. And please do watch the first video so that you can connect the Calvin cycle with the light reactions of photosynthesis. And also please like and share so that we could reach more students, especially the biology students, so that they can learn and understand more about photosynthesis. So always stay safe and keep up the good work. Goodbye everybody.